Good afternoon, everyone, and welcome to today's webinar co-organized by A&T Neuro. My name is Patrick Fiedler, and I'm Project and Product Manager for the EEG products at A&T Neuro and Imagine Medical Imaging Solutions. And we are very happy to be hosting this session today, and it's my absolute pleasure to introduce today's talk by Professor Jens Haueisen. As I mentioned, Professor Haueisen is the head of the Institute of Biomedical Engineering in Ilmenau at the University of Technology in Ilmenau. Professor Haueisen has been a Ph.D. student at the University Clinics in Jena, Germany, as well as the University of Washington in Seattle, USA. He has received a Ph.D. from the Technical University of Ilmenau for his thesis, Methods of Numerical Field Calculation for Neuromagnetic Source Localization. Professor Haueisen has been head of the Biomagnetic Center at the University Clinics Jena, and since 2005, he is full professor and head of the Institute of Biomedical Engineering at the Technical University Ilmenau. Jens has been co-authoring or authoring more than 230 peer-reviewed articles in scientific journals, working in the areas of measurement, modeling, and simulation of bioelectric, biomagnetic phenomena, neuromodulation, as well as ophthalmology. Today, Professor Haueisen's talk is entitled High-Density EEG with Dry Electrodes. Jens, thank you very much for accepting our invitation and being with us today. The screen and the stage is yours, and we are very much looking forward for your talk.
Thank you very much, Patrick, for the very nice and long introduction. I'm happy to be here today and talk about high-density EEG with dry electrodes. And I'm very happy that A&T is providing this series of seminars, and we have the chance today to actually provide some of our latest results or some of our statements about dry electrodes in general. So I would like to start, of course, with a general introduction to EEG, which is the measurement of electric potential differences on the head, typically caused by a current configuration of whatsoever type. It might be just a single dipole as here on the screen in yellow, or a much more complex distribution, which leads to some potential differences which we would like to measure. And the discovery of EEG goes back to the person here on the right. His name is Hans Berger, and he was a professor for neurology and psychiatry in Jena and discovered the EEG about 100 years ago in the early 1920s. You see his laboratory equipment on the left side, which is sort of bulky for the EEG devices we have nowadays, but he was using back then already electrodes, very similar type as we do here. He was experimenting a lot with different materials for electrodes and actually introduced the very first silver and silver-chloride electrodes, as well as gold electrodes, and amongst many other types. He was using most of the time electrodes which would stick underneath the skin. So these electrodes you see here, those are actually silver electrodes, and they would be placed underneath the skin of the patient, such that you would record more or less from the skull and not from the scalp. This is, of course, nowadays something we don't do anymore. You see the typical situation in a lab here. The classical EEG cap will be placed here. Electrodes will be filled with gel. And that procedure done here by the PhD students is very time consuming and requires quite a bit of training, as those of you are knowing who are within the field. And depending on the amount of channels, it can be easily up to even 1 hour. And that is not the only drawback. The second drawback you have, besides the long preparation time, is that you need skilled personnel for that, and you are not really able to do this outside the lab. So whatever type of experiment you're planning, be it like in this case a visual evoked potential, study or something else, you are bound to the lab or a clinic or similar. Now, what we would like to do is, of course, improve on this so that we will be able to get at the end a much happier face also with the PhD student. In this case, she's not very happy about the situation which is currently visible on her hair. And that is something which we definitely would like to improve. And you see how we do this in the next slide, in the next video. So actually we went here to the home of the PhD student. In her living room, we now see that she actually applies the cap by herself. So there is not really the need for trained personnel anymore. And the next thing to observe is that she is able to do recordings in a much faster or finished preparation in a much faster way compared to the wet electrode. So you just place the cup, move the cup a bit, and then start recording whatever you want to do in terms of your paradigm. This opens the door not only for much faster EEG recordings, it opens also the door for telemedicine applications relying on EEG. So you can think of many different aspects where you want to monitor patients at home or in some other remote place and have the analysis of the EEG / the interpretation of the EEG done at a completely different place. Now, finally, when you observe the video now, at the end of recording, there is no hair washing. You just place the cap back, in this case on the glass head or wherever you like, and you go and do whatever you want. So this is the big advantage in terms of the dry electrode recording. We will have a closer look at some of the basic requirements for such dry electrodes. We have grouped these requirements here in three main groups, which are the applicability, the reliability, and the producibility. And those include several subtopics, and we will touch in the talk a few of these subtopics. The 30 minutes are not enough to go through all of the details, but when you see, for example, biocompatibility, something which we definitely want to have because it's required by law, this, of course, is a material point. That means coating material of electrodes as well as substrate material of electrodes as well as the cap system has some important role to play in this respect. For the passing to the hair layer, we of course need to make sure that the substrate material has a decent material type / material shape, and the cap is supporting this, and so on. You can think of all of the different aspects here. Producibility is an important issue and is something where especially economic aspects play a big role. At the end, a cap and a set of dry electrodes needs to be affordable. Now, having worked on this field for a long while, we have tested many different types of electrode materials and many different shapes of electrodes and so on. And I would like to give you in this slide a short overview on what type of electrodes you might think of / you might have been using already. And the first thing to observe up here is that we have different materials. So there is metallic material like titanium and coating with titanium nitride, but there could be also plastic materials like polyurethane and titanium nitride coating. There could be gold pins, that is also an option, of course, gold electrodes. And one thing which I will actually outline in this talk more in detail is polyurethane as a base material and a silver chloride coating. This way, you end up with electrodes very similar to the silver-chloride electrodes you might know from your wet recordings. One other thing I would like to point out is the lowest line here, where we have the different weights of the electrodes. And one thing which is sticking out here, although such gold pin electrodes are easy to produce and actually not very expensive at the end, they have a big disadvantage. They are heavy if it comes to high density recordings. So imagine this 2.2 grams multiplied by 256. This is something which is bearable, but not very nice, as you can imagine. And this is one of the reasons why we went actually for a material which is polyurethane. The second reason is actually, and that holds similarly also to the titanium, the issue of hardness. When you have a metal pin electrode on your head, it's very refreshing and nice in the first place because it's typically a bit cold. But then after a while, it really starts being uncomfortable and in some cases even hurting. And in order to avoid this, you need to have electrode materials which are soft to some extent. And polyurethane is a material where you can tailor softness quite easily, such that indeed we are able to produce also soft electrodes. Now, with this final introduction slide, I would like to make 2 points. First of all, you can see here the first poll. Our organizers asked me to provide a set of questions for the poll. And the first question which came into my mind was actually that I would like to know, what is your experience in the field? Are you new to the field, have little experience in the EEG? Or are you experienced with wet EEG, for example, or even experienced already with dry EEG? So please let us know, and yeah, we'll see the poll results hopefully in a short while. Second point on this cap is that caps are important as well, and you see different types of caps indicated here, different cap materials.
Yes. Sorry to interrupt you. The poll is ongoing, so the users see the poll at the moment and not your slides. And we would ask everyone to select the corresponding options. Let's wait a few seconds.
Sure. I thought it's only in the chat that you see the poll.
No, it's in parallel. We switch either between the slides or the poll questions. Okay. So here we have the results now, and around 44% answered they are new to the field of EEG and have little experience with it. 47% answered they have experience with wet EEG, and already about 19% answered that they have experience or prior experience with dry EEG. And that's quite a lot, right?
Yeah, that's quite a lot. I'm very happy to hear. Yeah, thank you very much, Patrick, for the poll results. Yeah, so I would like to continue, and I'd like to point out that we have here different cap materials. I will not touch the cap materials today, but they are equally important when it comes to a good holding of the electrodes. With this, I would like to turn to a few results which I collected from some studies we did in the past, and I would like to give some, say, initial statements about the performance of these dry electrodes in comparison to wet electrodes. And the first result I would like to show you is a result from a comparison of two caps, 120 channel wet cap and a 97 channel dry cap. It's actually the first prototype we built ever with this type of silver-chloride polyurethane electrodes. It's the one you can see here on the right side. And with this, we did sequential recordings of visual evoked potentials, certain artifacts like eye blinking, and of course also eyes open, eyes closed in 20 volunteers. And this is what you see here as a main result for this eye open, eyes closed paradigm. Namely, you see the alpha band activity in a relaxed state for the subjects, and you see two lines, a orange one for the dry and a blue one for the wet. And you see the paraspectral density and the increase in the alpha band as a grand average across all subjects. And you see the nice alignment of the two curves, the wet and dry recording. Same holds actually also for the standard deviation, which you see as dotted lines here. There is a slight difference I would like to point out in the low frequency band, where the wet and dry give you slightly different results. And that is something which is noticeable also in other studies. Now, one thing we observed in this very first study was that contact pressure is an issue with the dry electrodes. That means if you have electrodes which are sort of loose on your head, so they're not really pressing on the surface with some force of whatever kind, then the recording is typically not at all, or at least quite noisy. And that means we have to look into contact pressure. And that is something which we did here in a study in 2018, where you see on the y-axis, the impedances here on the left side indicated, and you see that we changed the normal force, and we did this for just placing the electrode and then applying 1, 2, 3, 4 Newton. You see actually on the upper right the device for doing that, and subject having here this on the frontal position, and we also did, this is in blue, the temporal position. And we did different shore hardnesses, that is the hardness of the electrode. So a 98 is quite hard, a 70 is relatively soft. And what you can see here is that we have a decrease in these box plots for our impedances from 0 to 1, of course, but also from 1 to 2. But then from 2 to 3 to 4, it does not change much. So from this, we concluded that about 2 to 3 Newton are sufficient, but they have to be applied. And that is something the cap or the cap system has to provide. On the right side, you see a discomfort scale, because when you increase pressure, you increase discomfort. And that means here on a scale from 1 to 10, 1 is I don't feel anything, 10 up there is this is maximum pain, take it off right now. We are in the range between 2 to 3 to 4, that is the typical range for the normal, for the volunteers in this study, where how they rated the pressure in the different forces. Now, with this, it is quite clear that we need to distribute the forces also. And this ultimately led us also to a few changes in the electrode designs. And that is what you can see here. You see here 256 channel cap, which we produced about also 2 years ago or so. And what you can see here is a set of electrodes which are in the frontal part, having no long pins around the forehead, basically. And then we have medium-sized pin and then long pins. You can see the separate electrode here is also on the right side. And that makes sure that especially in the front part, where you're most susceptible for pain, your electrodes are not as painful. The second step is, of course, to reduce softness, reduce hardness and make the electrodes in the front softer, which also improves comfort in this situation. With this, we also did a comparison study, and this time I show you the results for the visual evoked potentials. You see up here the averaged about 300 trials per volunteer, averaged channel plot and butterfly plot of all the 256 channels for the wet on the left side and for the dry on the right side. You see the global feed power here underneath, and then you see for two of the main peaks here the distribution, and you can see that those distributions are very much alike. Please keep in mind that those recordings were done sequentially. So there was first one cap and then another cap, and then you have some time in between, so you don't expect it to be exactly identical. Yeah, with this, I think I would like to close this part of the talk, which is here summed up with, first of all, comparable signal quality for wet and dry recordings. The experience we have with this is now altogether beyond 100 recordings, beyond 100 volunteers being measured in such a comparison of wet and dry. Multi-pin electrode design enables the passing through the hair layer with some restrictions. If you have very thick hair, then you might need longer pins. That is something which we're working on right now. Dry electrodes allow for rapid application. I think that does not need to be any further, there needs no further explanation for that. All of our volunteers typically say that wearing comfort altogether across the entire session is better for the dry compared to the wet cap. With this, I would like to turn to another poll, and this is because the organizers also asked me to come up maybe with some funny questions. So here's a funny question. What do you see on this picture? The first thing is these are like fingers here in the middle, right? So is it fingers on a pottery wheel? Or maybe this is a sort of a butter cake and this is a clump on a butter cake. Actually, when I had this picture on the screen, my daughter showed up and said, well, Daddy, did you photograph toilets? So maybe it's a brush in a toilet. And finally, is it an electrode on the skin? So what is your suggestion?
Okay, thank you, Jens. The poll is ongoing. At the moment, around 50% of the participants voted. Let's wait a few seconds longer. Okay, and here are the results. So 11% answered it's a finger on a pottery wheel. 2% answered it's a clump on butter. 5% answered it's a brush in the toilet. And already 82% answered it's an electrode on skin. Is that what you expected, Jens?
Yes, that was my hope. Yeah, so it's electrode on skin. And skin in this case is actually artificial skin. So what you see is indeed a rotating disc. It does about 23 revolutions or rotations per minute. And this disc is used for testing the wear off of the electrode. So what you see here is a holder and an electrode. So you see actually the fingers of the electrode being, it's a soft one, being a bit tilted. And that is because there is a pressure of about 3 Newton pressing the electrode onto that rotating artificial skin. The artificial skin is silicon-based material, which is used otherwise. We just adapted it from the literature. And we use this here to actually do testing for the stability of the coating. And this is a thermography picture. And the reason why we did this actually was to see how much temperature increase do we have when rotating this disc. So the temperature would be an additional cofactor for the wear off. In this case, the temperature height here is 24 degrees roughly, and the room temperature is about 22 degrees. So we have about 2 degrees of increase. And that was done actually in order to adjust for speed. We typically do a few thousand rotations, and that means you have to wait longer if you do it slower. I want to show you another set of pictures for that type of approach. You see scanning electrode microscope photographs here of 3 different electrodes, an unused electrode, and this is actually always the pin tip, what you see here. Consider that this is 5 micrometers. This unused electrode was later on then having 1,600 rounds of the artificial skin, and you see the result is still pretty good in terms of coating. You have pretty much no differences here. And we have here another electrode after 800 rubbings on hairy skin. It's actually a male 4-arm skin where we move the electrode up and down. And we did this by now for 9 samples with the movement on artificial skin as well as on hairy skin, on real skin. And I can tell you that at least about 2,800 times 15 centimeter is doable, and you still have coating on your pins, and you still have contact that means resistance below 2 ohm. In most cases, actually, it's 3,200 rounds or 3,200 times moving up and down. You can think of it moving it up and down 3,200 times. You don't want to do it in one round, so we spread it over a few days actually. So with this, I would like to come to one other topic, the final topic for today's talk, which is the aspect of spherical harmonic analysis. And the reason for this is that I have with dry electrodes one issue which is really different from wet electrodes, and that is the mechanical buffering of the electrode gel or paste. That provides sort of motion buffering. So that means when your electrode is slightly moving because of head movements or something else, this gel typically provides some sort of buffering, mechanical buffering. And that means that we have more movement artifacts in dry electrodes compared to wet electrodes. So what can you do against this? Of course, you can use your typical signal analysis portfolio to go against the movement artifacts. But that sometimes is not sufficient. And in case when you have, for example, channel dropouts, you might want also to do a dropout correction. And this is what we do here with a technique which is called SPHARA, spatial harmonic analysis. The basic idea is quite simple. We do what you know from a standard Fourier analysis in spatial domain, in the EEG spatial domain, in the sensor domain, basically. The difficulty is that our EEG sensors are not like the typical image grid, so they're not regular. And in order to actually go for that, we do an eigenanalysis of the discrete Laplace Beltrami operator. With this, you can do many things. You decompose into basis functions, and those basis functions are orthonormal, and that means you can do at the end quite a number of different things with SPHARA applied. Spatial analysis, spatial filtering. You can do online filtering because it's extremely fast. It's in the few milliseconds range that you do the filtering. You can do dropout analysis and correction, and this is what I will demonstrate in the next few slides. And you can do data compression, but I will not touch on this last point here. So we have now the set of basis function as the main operating means for SPHARA. And you can see this here on the slide. We have our cap in the lower in the upper left corner, and the triangulation of that cap right next to it. And based on the same triangulation, you see the different basis functions with abbreviation BF1 to BF11 here in this case, but it of course continues. We basically have a set of basis function representing each time slice of our recorded EEG. So whatever it is, you can represent it in those basis functions. And with this, you can do filtering, spatial filtering. So don't mix it up with temporal filtering, right? This is spatial filtering. And the thing which you see here as an example on the upper trace is a single trial data set, which is actually single trial data from a visual evoked potential recording. And you see that we have some noisy data here. And after the spatial filtering, so time step by time step, we have this filter result down here. So it takes away quite a bit of the noisy data, as you can see here. And the same thing, so we do the single trial spatial filtering and then average afterwards. And the result you see for the VEP is indicated for the subject here. So you see the more noisy data without SPHARA filtering, without spatial filtering in the upper part, and with spatial filtering in the lower part. Consider there is no temporal filtering in this comparison. It's just spatial filtering, right? And you see also the change in the temporal properties. The curve, when you, for example, look at the blue curve, it gets much smoother, for example. And last but not least, I would like to show you on this slide a example of channel dropout correction. So what you can see here in the upper part is a visual evoked potential signal. You can see the butterfly plot on the left and the topography here in the middle. And this topography is for the maximum here, of course, indicated. This topography we take and we simulate a channel dropout of altogether 19 different channels. You can see those 19 channels indicated in the lower panel here. And those channels are actually continuous. That means it's really a patch of channels. Now we use our SPHARA basis functions to reconstruct what those channels would look like. And you see actually the result as the topography map, the color map underneath. And you see that there is a slight difference when you look at the minimum value here versus the minimum value here, but altogether the reconstruction of this topography here is quite well performed. Same is holding actually also for the interpolated butterfly plot, which you can see here on the right side. So with this, I would like to end for the scientific part. We have by now achieved with our dry electrodes quite a number of important steps. They are biocompatible. We have decent signal quality. We find a very nice situation in terms of electronics compatibility, but of course there are open issues. Contact stability in terms of caps is still something we're working on. Patient comfort is not always perfect. Fitting of the electrodes is not always perfect, and costs are also not yet there where we want to have them. But this is ongoing work, maybe for the next talk. Outlook, we will have improvements in wearing comfort. This is what I can promise already. We're working on this at the moment. You can do already with the 256 cap, as you can see here on this lower picture, measurements in supine position. That is important, for example, if you want to go to fMRI. And that is something which, of course, with the current electrodes will work, but we will improve on this as well. And last but not least, we will have modular and adaptable caps, which are better suitable for the different head shapes we have, and adjustable for the different hair types which we have. And this, with this, I would like to end, and thank you very much for your attention, and also thank my collaborators, my PhD students and postdocs, and our organizers for this webinar, and last but not least, of course, for our financial support. And as this webinar is recorded and will be made available later on YouTube, I have on the last slides a bit of literature to read if you wish. So there's some on the dry EEG, there's some on dry EEG and TES combination, there's something on SPHARA, something on the electrode technology as such, and of course something on applications. I picked here sports science applications because we have a good collaboration with colleagues in Italy, Professor Comani, Professor Bertello, etc., which I would like to mention here. Yeah, thank you very much for this, and I'm happy to answer your questions.
Thanks a lot, Jens. That really was an excellent presentation again, and very informative. I hope the audience enjoyed it just as much as I did.